Welcome back to Office Hours Unplugged, where we chat with the most prominent researchers and PIs here at McMaster. Today we bring with us to the studio Dr. Andrew McArthur. Could you please introduce yourself a little bit to the audience and let them know a little bit about yourself?
Sure. I'm Andrew McArthur. I'm a professor in the Department of Biochemistry and Biomedical Sciences, so Faculty Health Science. And I'm also in the Michael G. Grudy Institute for Infectious Disease Research. I know we're going to get in the career. I've had a very nonlinear career, but my specialty is computational biology. I work on surveillance of infectious pathogens, but from the data perspective. Yeah.
Awesome. So just to get us started, how exactly did your journey in academia start? Was it something you've always been interested in, or is it just something you came across during your time in undergrad?
Yeah, so no, there was never a plan. I'm not one of those profs that always was in mind. Honestly, I went to my undergraduate because I knew education was the way to get out of town and do other things, but
Right.
I didn't really have a plan. I did enjoy the curiosity. I did enjoy research, so I decided to go to graduate school. Ended up doing a PhD, but certainly wasn't dedicated to an academic life. I still was interested in postdocs, but I thought industry or government service. And in fact, I did leave academia and did various things before returning to academia 10 years ago at McMaster. But no, I'll readily admit a large part of my career, I was very mercenary. I had a good set of skills, DNA and genomics and sequencing and computational, and I went wherever people were willing to pay me to do interesting things. It wasn't really till I came to Mac 10 years ago that I had a mission and a project in mind, something to make a long-term difference.
Yeah, and that's it's very interesting you mentioned that. What brought you back into academia after such a long break?
Yeah, so I my first sort of stage of the career, Canadian, and did my PhD here. Went to the Smithsonian in the United States around DNA sequencing technologies, but really was frustrated at the time of how little data you could generate. I wanted to get involved with genome projects, which were at very early stages. And the big ones I went to a place called the Marine Biological Lab. They were looking at parasites, particularly protists and parasites from an infectious disease point of view, but also evolutionary biology. They're all ancient, but they were doing amongst the first shotgun genome sequencing projects outside of bacteria.
Wow.
And so I developed, and I had an American lab and built that. And then the housing crisis happened in 2008, and all the U.S. fell apart. My wife got an offer here. I retired, and I raised kids for 10 years.
Wow.
But in that window, I formed a company. And my company did a lot of work with Mac, particularly around antimicrobial resistance, and Mac made me an offer. And it was just a timing. It wasn't a plan, but Mac and what was going on in my skill set was the right time at the right place to do a difference. Biggest driver was, in industry, you don't have access to students, right? And that was a big driver. The more I thought about it, the more it'd be great to have students, right? And do it from that perspective.
You mentioned AMR, and I know it's like a big part of your lab and your work. So what was like your first interaction or your first, I guess, time you kind of saw what AMR is and its importance?
Yeah, so I had my company doing consulting around genomics and data science and DNA sequencing. Literally was not in the AMR space, though I'd done some work for Public Health Ontario. I got a call from a colleague, said, hey, will you sit in a meeting at McMaster? There's some Canadian researchers and British researchers getting together thinking about database approaches in genomics, and they really could just have someone in the room who knew this stuff. And I didn't know, I knew about AMR in the broad sense, but nothing specific. And out of that, my company, started doing some work, started doing some pro bono work, and then led to that. And it became that effort needed a champion, and
Right.
I was the right guy. But it was early days. In that meeting, there was a deputy minister from the UK, and he privately announced that there were completely drug-resistant strains of gonorrhea in a high school in UK. That was that day that was announced, and
Right.
now that was globally. Those are in our community. So that was really a change that I had the company as goals. I really came in to do pro bono and give them advice, and then realized, oh, wait a minute, I could actually make a difference here, right? And so started working with the folks at McMaster.
That's amazing.
And how did you kind of get sucked into computational biology? I don't know if you talked about that.
Yeah.
How did you kind of funnel into that from a background in science?
Yeah, so like my PhD was in deep sea oceanography and paleontology, picked up sequencing there. The only commonality in my career is computation. So my godfather gave me my first computer at the age of 9 in the 80s.
Wow.
And yeah, I was an addict from then. I'll readily admit I was a childhood hacker. And that was the one consistency. And being that early generation, and then the PCR got invented, Sanger sequencing became larger scale, and then we moved into pyro sequencing. Often I was the only one in the room with the skills, the computing skills, because we were generating big data. And honestly, that was the business model for my company for 10 years.
Yeah.
The equipment, the ability to generate huge data was well ahead of people's ability to handle it. And I was one of a handful, right? And so a lot of the timing. But yeah, like even when I got my PhD, my advisor said on the day of the defense, like that, drop that computation stuff. There's no future in it, right? Her advisor, who was in his 90s, said, no, that's not true. Stick with the computation stuff, right?
Yeah.
It wasn't until I came to Mac that, well, my company first, but then Mac, where I was 100% compute. Like even my lab in the U.S. had cell culture sequencing, molecular biology, but the world has changed a lot, and so a lot of it's timing. But a self-taught hacker, not a proper software engineer. Yeah.
There must have been like many difficult parts in your journey, especially running the business and then going back into academia. Is there any like notable challenging moments that really defined who you are in your career?
Yeah, so these careers are extremely rewarding, have no doubt. They're not easy, they're long, and can be a grind aspect to it. I would say the toughest was my American years. I had a lab in the U.S. system for about 10 years, so unlike here, they're called a soft money. I was at the extreme, so if I didn't raise a grant, we wouldn't have heat, electricity in the lab, but neither would I get a paycheck, right? Or if I screwed up on a grant, then someone lost a job because I couldn't pay for them, right? So those were the hard point. I was working 70 hours a week. It made my career, have no doubt. At the time, I was one of a handful of Canadians that had worked at that level of work. I was surrounded by 14 Nobel laureates.
Wow.
But I'll tell you, I don't remember my second child's first year of life. I know I was parenting and up at night, but I was so fried from that. And then the stress was when the U.S. system in 2008, hopefully not again, but the housing crisis fell apart in granting, and I had to make a change, right? So that's a pretty extreme, but even now, if you look in the Canadian system, funding rates are extremely competitive right now. So profs are not getting grants at the same rate, right? While we're trying to raise stipends to pay for our students, and so everything gets more expensive. It's another tough time. So it's about that persistence. Do it. In some ways, having the company was a luxury. I stepped out of the academic challenges because it's an all-in enterprise. Hugely rewarding, but takes work all the time. Companies are honestly more relaxed.
I was actually gonna ask you about that. I mean, what was the standout difference between managing a company and then managing a lab? I mean, what was the difference in workload and how you managed your the people working in your lab or your company? What was the differences there?
Yeah, so I originally, when Joanna got the offer, my spouse got the offer here to Mac, and we moved back to Canada, which was great, and to Hamilton, which was great. Our family's from here. My ancestors are former mayor. So I originally was a stay-at-home dad. That was the plan. I didn't plan on a company. It's just because I'd done things that others done, the phone kept ringing. So the company didn't start out at full throttle. It started out when the kids were napping type of thing. And then as they got older, taking on more and more. And most of my clients were American to start and then spreading out in the Canadian. And honestly, the business model had a huge component of community service. So I did a lot of community work. That was a struggle to maintain when I came back to the university, though you'd think working with undergrads is a form of community work.
I would say so,
the company was designed to scale. There was a deeper part too. So we come up in the U.S. system. The MIT report had come out on how women in the early of their careers, basically everything's tipped against them, right? They get promoted less, they get lower starting salary. So my wife got the offer here, tenure track position. I said, I'm going to be a stay-at-home until you get tenure. We're going to tip that table in your favor, right? And then I loved it and didn't stop, right? And so there was also a decision on that. So the company had, even though I wanted to do the science, I wanted to bring in income, I wanted to keep my reputation and publications going.
Right.
That was secondary to community work and family life, right? So by the end, that company was almost full-time, but honestly, it was getting tough to maintain. Sequencing and that had become such large scale. I was spending more money on hard drives than I was on taking salary, right? It was, and so the company probably didn't have a lot left to it. It probably had to merge with a larger firm, so, but very different, right, perspective. Yeah.
And one of the more interesting parts of your career, I think, you talked a lot about it in your guest lectures, and I think it would be really great if you can kind of talk to the audience about the role your lab played during COVID-19.
Yeah.
I remember listening to what you were saying and all the amazing, commitments and, the sacrifices you had to make. But at the end, the results and the outcome was really amazing. So could you kind of let the audience know a little bit about, the contributions you made?
Yeah, it's I guess you get what you train for,
right,
in some sense. So the advantage of the Canadian system, the community that works on epidemiology of infectious agents is quite small. Includes government and academia that work quite together. So before even the first cases in Canada, we were talking and we were planning, right? And so there's a real advantage to that. In Ontario, what the case was, the provincial labs just did not have the capacity for variant tracking, right? They'd been underfunded, whereas Alberta was ready to rock, right? BC was ready to rock.
Right.
So Ontario Institute for Cancer Research and McMaster stepped up in the early days of sequencing capacities, and Mac gets a lot of credit. I called the VP and said, look, we need to track variants now. I have no money. And they just said, get started. We'll figure it out later, right? And we did figure it out later. So that our initial year, we were working with 26 hospital systems. Those daily reports of what variant was where was us with our colleagues.
Wow.
And we had to build a sequencing team from scratch, a bioinformatics team from scratch. Out of that, we also wrote the national software for when you sequence a nasal swab, get that raw data predicting exactly what variant is in there. So we made the national standard. Really proud of that. What was the difference being here, surrounded by infectious disease people? Our software doesn't do anything particularly different from other platforms that people wrote, but it was written from a pandemic perspective. It was for scale. Right. So it runs at least 10 times faster than any other point, and it can handle, therefore, thousands of more samples. Wow. Because early days when we had 2 patients, right, we said, give us a couple months and we'll have 10,000 a day. So it was pandemic software, which is pretty cool to it. So by the end of the first year, we could retire a sequencing team. The capacity was local hospitals, but for years, until 6 months ago, we were part of the weekly reporting, say, of Halton. We were part of the team that designated Hamilton as a Delta hotspot, which means vaccines came early here. So invariably, we probably saved some lives from that perspective. A lot of awful stories too, obviously.
Right.
We lost a lot of people. I don't know if I ever want to do one again. Those were 20-hour days for long, right?
Wow.
But I come from a military and other family. Service is really important. None of us, my generation, went to join this, but this was my calling. This was my turn, right, to serve. And my lab, I say but it was all my students and staff, doing this. So it was extraordinary, right? And even now we're maintaining the software platforms being used around the world to track variants. But we're, thank goodness, not doing the lab work anymore.
It must have been very satisfying feeling to see your work be utilized immediately.
Yeah, and the pressure, those early days, those diagnostic tests, the virus was mutating at the priming sites for PCR, so the diagnostics were giving false negatives.
Oh.
So we were involved in that, working with companies saying we got to redesign the kit because the variants now are giving false negatives. And then obviously the real concern was, how's this thing going to evolve, right? And transmissibility, no question, right? We were just, we got lucky that it wasn't lethality, right? That really increased.
Were there any significant lessons you learned from the your experience with COVID-19? God forbid there's another pandemic. Is there anything you'd recommend the government to do or any other researchers to do?
Yeah, so I think one of the things I learned is the different roles that public health versus academia apply. In this casing, we threw out all those roles and we integrated. And really, an academic should not be embedded in the provincial reporting system. There's ISO certifications, all the rest, and so we had to learn that. It really reinforced what students do. A lot of our work was done by a third-year BDC student. The academic ability, that training and how students think, the ability to adapt and develop new things, and remarkably quickly. Academics do not have a reputation for being quick. That was really sunk in. But it also was obviously we're not trained to do the daily counting. That's really what public health officials are doing good at, making policy decisions. So I learned where our limits are, I think was a really important thing, and where we could focus in. So our existing research program, it's really helped to target, okay, let's do where we can make a difference and not bleed over. Let's collaborate with the other people that do it as opposed to doing it ourselves, right? Yep.
So you do agree with maybe that there are pros to the integration between the academic sort of side of things with labs and the public health officials. You think that this is something that would be beneficial to people if it continues in the future?
Yeah, like the U.S., it's setting aside the current status of the government there, but traditionally in the U.S., it's hard for academia to work with federal agencies and public health. That's very much not the case here. I know, we work closely with federal scientists all the time. That integrated system was a huge benefit to Canada, and working closely with them helps identify their needs, right? And we can, and it might be building something because we're engineers, but also could be pure research, like we don't understand something. And whereas government has to generate data to guide policy, right? And so we've learned on that. I do, a lot of my students work with government policy, so they understand, so they see that useful endpoint, and then vice versa. There's young government scientists that come and train with us to learn the research perspective. It's a pretty lucky place. It's why we did as well as we did in the pandemic, because of that attitude. There's issues with data sharing in Canada. Canada's famous for having data sharing struggles between provinces and federal. Netherlands and UK did so much better than we did. We learned some of those bigger issues too that we need to work on.
You mentioned like Netherlands and UK. So like, do you think there's anything we can learn, like as Canadians and as researchers, like from the Europe's policies and how they implement certain acts and laws?
Yeah, I think data sharing is a big one, right? So like if you're sequencing variants, you do need to connect it with the clinical or patient data to figure out, is there a trend, right? Is it taking off in immunocompromised patients? Well, if you don't know who's immunocompromised, you can't figure that out. That's our federated system has not really got up to that. UK internally and the EU internally have really spent a lot of effort. What came out of it, though, at the genomic level, there was an international data sharing effort, and I would sit and read the report. So there were times where, we're on Zoom with Denmark or Netherlands, UK, because we're seeing the same variants causing the same problems, right? And we start coordinating it to it. The UK did better, right? Sweden has a very integrated health infrastructure system, so it helps easier to connect outcomes to genomic variant. That's really hard in Canada. I think a lot of good federal recommendations are coming, but we are a federated country, right, of provinces, territories, and so there's things that's broader issues to work on. Yeah. But a lot of our colleagues in social sciences here, like that's their expertise. They can make those contributions. Right?
Well, zooming out a little bit from, I guess, SARS-CoV-2 and infectious disease stuff, can you tell us a little bit more about your work with AMR and your contributions to the things like the Comprehensive Antibiotic Resistance Database?
Yeah. So, like in contrast to COVID, we knew this would be a fast pandemic, right?
Yeah.
We were also fairly optimistic. We were worried that what mortality rates and hospitalization rates would be, but we had those genomes early. We expected vaccines. mRNA vaccines have been underdeveloped for just that purpose, even though originally they're designed for fighting cancer. AMR, antimicrobial resistance, so that's when an antibiotic fails and your infection can't be treated. This is a slow pandemic, so every decade it gets worse, and it is decadal to win. And unlike COVID, we generally don't get good outcomes. It gets worse, right? So, in Canada, well over 20% of cases fail their first treatment, right? We routinely have untreatable, like as I said, there's untreatable gonorrhea out there, right? Tuberculosis in the North, Canada's got some of the worst rates. So we are basically looking at the loss of antibiotics, which you think of that like people will die, but also you can't replace your grandmother's hip if you can't do secondary control of infection. Right. You can't do chemotherapy, right, because you're suppressing the immune system. All modern medicine relies on this. The outlook is not good, right? There are efforts to maintain our antibiotic arsenal, but there's very serious efforts on what comes next when we lose the antibiotics, whether they be phage viruses or adjuvant molecules. One of the big aspects is we need to know where the AMR genes and pathogens are and how they move about and which ones really lead to bad outcome for people or animals. Because remember, we use antibiotics for animals and aquaculture as well. So that technology is genomic. That's why we were ready for SARS. We were already sequencing bacteria from farm, from wastewater, from people, from swabs. My group essentially builds that infrastructure. So when you sequence an infection, we have a database that in our software and other people's software to say, okay, that swab you just sequenced that has whatever pathogen in it, the reason these drugs are failing are these type of genes, right? And then we help researchers fill those gaps when we don't understand. We also license to companies. So, and say a cute one is a brewery in Japan. They want to make sure their vets are not growing bacteria with AMR genes, right? That's part of their regulatory environment. So they sequence their vets and use our software, right? Probiotics. You don't want probiotics first to that. So we are very much engineers. We build software and databases for surveillance of the genes and mutations that cause antibiotic failure, and then we deploy them to companies, public health agencies, researchers, whoever needs it, right? And then when needed, we get directly involved, with individual patients or communities. Yeah.
And correct me if I'm wrong, but like one of the reasons AMR has become such a problem is because during the 90s and like during like the first kind of creations of like the first antibiotics, people were really using them for everything. Right, like they were just using them a lot, and is that the reason why AMR has become such an issue? Like the overusage, or maybe even the unnecessary usage of antibiotics.
Right, or the misusage is a big one. So overusage first, like in the Second World War, we used sulfonamides in the ton level to try to keep lung disorders down. Right.
Right.
At a level that we do not use antibiotics now. Misuse is a common one. So your doctor says, "Yeah, you feel better. Finish your antibiotics." That's because if you finish early because you feel better, you'll probably be fine, but you're going to breed drug resistance. You got to follow through and knock out those bacterial infections, right? If you overuse and constant exposure, bacteria will evolve. They will become resistant. So we have overused, misused antibiotics. We used to use antibiotics in agriculture as to promote growth, right? They had nothing to do with infection. You got fatter pigs.
Right.
But we were breeding resistance as a result. So that's illegal in most countries now, right? But yes, we've had decades. We had a long stretch where we just kept finding new antibiotics. So even if resistance, like penicillin, when it was discovered 3 weeks later, so was the first resistance, right? But the assumption was always fine. We get resistance, we're just going to find another antibiotic. Well, that hasn't been true since the late 80s. It's become very hard to find new antibiotics. Even when we do think we find a new variant antibiotic, give it less than a year before we see resistance, right? So the well has gone dry for finding new drugs while the resistance is increasing. So you're running out of antibiotics. If we were still discovering new molecules all the time, it's an arms race that we would be winning. But we're not finding new molecules. And so every time we make a mistake and overuse antibiotics, we lose molecules and we run out of options, right? And so it's an natural outcome. Bacteria use these molecules to talk and kill all the time. Right? And so they're primed for millennia to do this. So we really don't have a lot of advantages here.
Yeah.
In a way, using antibiotics should ultimately lead to the lack of use of antibiotics. We'll lose them. So am I optimistic that we will find solutions? Heck yes. Will that be quick? No. And will it get bad between now and then? Very much so. And you got to remember, we're sitting here in Hamilton. We got a great children's hospital. If you're in sub-Saharan Africa, right, or in your refugee camp, it's a whole different ballgame of the level of treatment you're going to get. It's not going to be an equitable thing of where it kills and where it undermines medicine. Yeah.
And do you think the solution to that would be to maybe upregulate or just increase the level of monitoring we have overall, or what would be the way to go about it?
Yeah, so Canadian framework has our response has 3 pillars. One is stewardship, so just be really careful in how you use your antibiotics, be smart about it. One is discovery, let's find some more, which is very, very hard. And the third is surveillance, what's going on. Part of that's phenotypic, just reporting like, this infection, the antibiotic didn't work. We count those regularly. And that gives a sense like, penicillin is dropping in effectiveness. But increasingly it's genomic surveillance, which pathogens, which genes. And you got to remember, we use antibiotics at the farm. Those meats go through an abattoir, they end up at the grocery store, they got people. We use the facilities, it goes out wastewater into rivers and natural environment. It's a complicated system. We don't have enough money to fight resistance at all those points. So we got to know where everything is to figure out where we can spend the money most effectively. And that's surveillance, and that's genomic surveillance. So that's ultimately what we're trying to do in Canada. It's very much a One Health approach, trying to find the weak links that we can attack most effectively.
And going back to your lab, like what was the original vision when you first created your lab? Like, was it because, you really saw the importance of surveillance, or was it because, you saw the threats of AMR? Like, was it a combination of those things?
Yeah, it's a really good question. So when I formed my lab in the States, I was interested in big genomics questions and playing with computers. Happened to be the money to do that was in African parasitology. So I worked on schistosomes and giardia. But was I fundamentally interested in those organisms? Not really. I like the methods, right? Very different from when I came here. My company was already working. We knew the level of the crisis. I had the depth of the experience in this world. The field, the genomic revolution was just starting to hit. And so, I came in with a 20-year plan. Ultimately, the plan was we would build database surveillance software, and after 20 years, someone would have solved AMR and I could turn it all off. That's not going to happen. But the goal is very different. We were evangelical the first 5 years, going to public health and saying, hey, if you sequence, this is what you can learn, and we'll build the tools for it. 5 years in, the sequencing was everywhere. So the next 5 years we have to make this super robust so they can trust the results and know the mutations. Now this next 5 years is trying to figure out risk. So if you see the gene, should you freak out or not, right? Or should you change treatment? Should you put out a community warning, right, about it, right? Very different that we came in. There was even not only the science, but the business. So in Canada, US, database approaches are inconsistently funded, and great tools get built and then they lose their grant and they disappear or they atrophy. So CARD, our database and everything, was designed from a business perspective as well as a grant. So we get grants from CHR and that, but that pays for the undergrads and grad students to do the brand new stuff. Break new knowledge, right? And on that, the day-to-day of keeping a tool available for public health and all that's done through commercialization. We license to companies. So how I designed the lab from day one was this hybrid of a business perspective and a researcher's perspective. Thank goodness I'd had those 10 years in industry. I knew how to do that. But again, the US experience, I built databases, and as soon as we put the pipetter down, the money disappeared and the database died. So I designed in part. So it's very different, an academic lab. I came late in the career to academia, so 20 years, to build something and really make a difference. We got to hit it hard day one, but it has to be sustainable long term. So we had a business model to it. So even for the graduate students who are doing really cool work, or the undergrad thesis students, breaking new knowledge, it's in that framework of the reason we got to break that knowledge is 2 years from now, public health is going to be using this approach. So we got to get ready for it, right? So it's very applied, I guess, in the end, right? As opposed to basic research.
You mentioned your graduate and undergraduate students breaking new knowledge. I assume they're taking on vastly different projects between all of them. Is there any project in specific that excites you or something you're interested in currently that your lab is exploring?
Yeah, I think my research really comes down to two things. There's the AMR problem of building tools and databases and doing surveillance work and with government. For any of that to work, what we do is what's called biocuration. We take knowledge and we turn it into data and algorithm. So you can do that work. It's an underappreciated art, but we're surrounded by great databases, NCBI and all the rest. We're interested in that art, like how do you reliably biocurate? So if you look at our databases now, we hand curate every data point. So the trust factor is super high, means public health can really trust on it. But also, it's not like I'm going to have money to hire 100 people, so we can only do so much. So where we're playing now is AI and natural language processing. So we have undergraduates and master's students that have written AIs to read all the scientific literature to pull out information that we just couldn't manage by hand. The research in there is figure out whether you can trust them, right? And whether that's reliable information. So the biggest one from the very first meeting, with the British and the Canadian, they said we need to know what's called the MIC, in other words, the dose at which the drug is failing. For all the different isolates in the world and all the different genes. At the time, I just laughed because I could see that's 1,000 people trying to read all those papers. Well, now I have an AI doing that, right?
Wow.
So 14 years later, we're finally at that initial request. But the research of figuring out how to build the AI, how to train the AI, and whether you can trust the AI, right? And then ultimately convert that into risk metrics that you can make decisions on. I'm really excited by that. So I love how it's going to impact AMR work and surveillance and public health, but I also love just the pure science of knowledge acquisition, right? And to combine human and computer assisted approaches. It's pretty cool. We're doing a lot of cool surveillance and wastewater, super expensive, very difficult. So we're working with lab people and we've made that method 1,000 times cheaper, right? And so we're also paying attention, it's great, you can build a database, but you still got to do the work, right? So we collaborate with people to say, okay, let's make enabling technologies to actually gather the data in the first place, right? So those are pretty exciting too.
So you mentioned collaborating with some other people in some other labs. Can you talk a little bit more about, the collaborations you're doing?
Yeah. Science is collaborative, right? And we talked about the difference between my American lab that had cell culture, molecular biology, sequence. You had to do it all yourself back in the 90s,
Right.
right? Early 2000s. It's not that world. So I have a data science lab, but if there's an outbreak at a hospital, I can contract for someone to get swabs, and then I can contract for someone to do DNA sequencing and send me the data, right? We also do that collaboratively. So one of the plums of coming to MAC, right? This is one of the leaders in infectious disease research. We have an open door policy. So as people doing bench work find something and they need data help, they walk in the door. But also if we use an AI and it makes a prediction, we can walk across the hall to a chemist, a molecular biologist, a microbiologist, and test our hypothesis to make sure, can we really trust all this code we just wrote, that type of thing. We also really special about MAC. MAC is the home of evidence-based medicine. We have some of the best clinical epidemiologists in the world. So even though I really came for the infectious disease researchers, it's been a real pleasure in 10 years that we work right at the front line with patients and clinician researchers, and we do studies in wards and apply these techniques to people who really know how to ask the really difficult questions in a clinical or population setting. So that's pretty heavy. And then nationally, it's what we do, our database, all that is a national consortium. There's researchers in almost every province and territory and federal government. They're all part of it. When we publish those papers, it's like 60 authors. And then it is we run this database every 4 minutes. Someone's running a superbug through our algorithms, so we collaborate internationally to it. It's that's one of the huge fun things of research in an academic setting, right? You don't ever attempt to do it all by yourself, right? And you even our software for COVID, one of our core developers was a black hole physicist. He knows nothing about infectious disease, but man, can he write code, right? And so we were glad to have him, right?
I will second that is honestly the most beautiful part of research, is the collaborative nature of everything. What would you say so far is really your proudest accomplishment in your career or your research?
Yeah, it's a tough one. Pride's a tough thing. So, yeah, I'm proud of our COVID work, right?
Yeah.
There was a need, and with Mac's support, we were able to fill it and answer a lot of questions, and a lot of good work came out of it, right? Knowledge. Never mind the knowledge, too. There's just capacity building. Students learned, and they're out there with that knowledge, so when the next one comes, they'll be ready, right, to apply that. I'm proud of what we've done with the drug resistance work. As I said, the fact that our algorithms get used every 4 minutes is pretty impressive. And really, I'm the supervisor. I'm not the one doing the work, right? It's really students that do that work. But even back, in the U.S., like we sequenced the Giardia genome. So Giardia is a waterborne pathogen. At the time, was the third largest killer of children internationally. Out of that genome, that I did the sequencing and was also the computational lead. New drugs have been found, and so we don't consider Giardia a top 10 killer of kids anymore, right? Wow. And so that meant that primary data that we, as lots of us, helped generate, really led to much better outcomes, right? And so, yeah, you take in that I have a strong service perspective. That's what I'm most proud of, when helping others. And yeah, the bigger picture is, I love going to convocation. I love watching all the students and what they've achieved. I know you guys hear that a lot too, but that actually is a huge thing for a prof, right? Yeah. To see people that come in really green in first year and see how they grow and develop and figure out their path, whether that path is researcher, industry, government, arts, you name it, right? These are all perfect. And so that's, I'm pretty proud of what we do overall here at MAC. Yeah.
That must be amazing, seeing people kind of take over the torch, and even if they don't necessarily do exactly what you're working on, still taking inspiration from your work and your experiences, and moving to different aspects and different careers. That must be really amazing.
Yeah, I think, like I always say, my lab's not designed to create more profs. It's not really our goal, though I'm sure we will make some. And my trainees end up everywhere. Right. In industry, government, completely different fields. There's only a minority that really want to stick in this pocket and work on that. Right. What we do is we take biochemists, life science students, and we add data science skills and computation, and they take them anywhere, right? And build their careers and build their families and do what they want. That's a wonderful thing.
Yeah. Well, I think we're going to wrap up here. Thank you so much, Dr. McArthur, for taking the time to share your story and your science. We really appreciate it, and we think the audience does as well. Thank you so much, everyone, for listening, and we'll see you next time on Office Hours Unplugged.
Perfect. Thanks, guys.